Hello friends, welcome to Sam Biology class. Friends, today we shall discuss about the structure and function of hemidesmosomes. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Hemidesmosomes are specialized junctional complexes that contribute to the attachment of epithelial cells to the underlying basement membrane. They are present in stratified and other complex epithelia such as the skin, the cornea, parts of the gastrointestinal and the respiratory tract. They are integrin mediated. Hemidesmosomes are also involved in signaling pathways such as keratinocyte migration or carcinoma cell intrusion. Ultrastructurally, hemidesmosomes display a tripartite structure having an outer and inner electron-dense hemidesmosomal plaques separated by a less dense zone. On the basis of their protein components, hemidesmosomes can be distinguished into two types, type 1 and type 2 hemidesmosomes. Type 1 hemidesmosomes are found in stratified and pseudostratified epithelia, for example, in the epidermis. Its major components are integrin alpha 6b4, plectin isoform 1A, tetraspanin CD151, bullous pemphigoid antigen 1 isoform E, also called BP230, and BPAG2, which is also called BP180. These are the major components of type 1 hemidesmosome. Type 2 hemidesmosomes are present in simple epithelia as that of the intestine. It consists of integrin alpha 6b4 and plectin. BP antigens are, however, absent. This figure shows the major components of type 1 hemidesmosome. This is the basal keratinocyte. Keratinocytes are the epidermal cells that produce keratin. The keratinocyte, which is present at the basal layer of the epidermis, is called basal keratinocyte. Immediately beneath the plasma membrane of the basal keratinocyte, there is an electron-lucent zone called lamina lucida and an electron-dense zone comprising closely packed fibrous network called lamina densa. These two zones, lamina lucida and lamina densa, constitute the basement membrane. So this figure shows as to how the basal keratinocyte is strongly attached to the basement membrane by hemidesmosome. The major components of type 1 hemidesmosome are integrin alpha 6b4, plectin, BP230, BP180 and CD151. CD151 is a member of the tetraspanin superfamily. It aids in hemidesmosome formation. Next component, integrin, is a heterodimer. It consists of a smaller alpha 6 subunit and a larger beta 4 subunit. It acts as receptor for laminin. Another component, that is BP230. Its main role is stability of the hemidesmosome. BP180 also associate with the laminin. Intermediate filaments assembled from keratin 5 and 14 associate with the inner hemidesmosomal plaque via binding to plectin and BP230. These two components form critical links with the transmembrane component of the hemidesmosome and form the outer hemidesmosomal plaque below the hemidesmosome and parallel to the plasma membrane. There is a thin electron dense line called subbasal dense plate, and you can see the presence of collagen type 7 molecules in the lamina densa. In the lamina densa, most of the type 7 collagen molecule form semicircular loop structure. The semicircular loop structures are called as anchoring fibril, which originate and terminate in the lamina densa. Lamina densa anchored fibril, after entering into the dermis, encircle or link with the collagen fibers and other molecules in the dermis and provide a strong anchorage. Next function of hemidesmosomes. Hemidesmosomes promote the adhesion of epidermal cells to the underlying basement membrane. They are also involved in signal transduction. For example, hemidesmosomes are involved in signaling pathways such as keratinocyte migration or carcinoma cell intrusion. So friends, this is all about the structure and function of hemidesmosome. For more update, please subscribe my YouTube channel Sam Biology Class, and also please share your comments. Thank you very much.